Are we ready? All right, let's begin then. Hi everyone. For those who do not know me, I'm Alessandro Scagnorsini, and I'm going to be presenting my work I've been doing with Dr. Gluckman and Dr. Adair for the past year and a half, more or less, and it's going to be very exciting. So we've been working on the realization of porous silica nanoparticles for analyte sensing in the brain. Everything starts from the action potential. The action potential is the basic unit of the brain, equivalent to the ones and zeros in the computer, but for our brain. And from this, everything starts, our thinking and all our processing, essentially. The action potential is thanks to a balance in ions in and outside of the cell. We're going to call the inside of the cell intracellular space and the outside of the cell extracellular space. As you can see from the small diagram on the right, the extracellular space is rich in sodium ions, while the intracellular space is rich in potassium ions. So when we are at the beginning of the action potential, these balances are shown in the diagram, like the following, essentially. When we are starting the first part of the excitation, what happens is that there are two different channels, ion-specific channels, on the membrane of the neuron, essentially. And we have a sodium-specific and potassium-specific channel. And at the beginning, what happens is that the sodium-specific channels, they have two gates, and both gates, they open, allowing an influx in ions inside of the cell, as shown by the arrow. Once we reach the peak in the action potential, the first of the gates closes, making the sodium channels enter in an inactivated state, where no influx of ions is allowed anymore. It is then the potassium channels that open and allow an efflux of potassium ions in the extracellular space, leading us to a lower membrane potential. Finally, now that we have our concentration kind of flipped, so we have many more ions outside, the potassium channel just closed, and we have a lot of sodium ions inside of the cells. What's going to happen is that we need to repolarize the membrane, and that is done thanks to the sodium-potassium pumps, which is activated by ATP, which is the product of the metabolic activity of the cell, thanks to oxygen, essentially. So as we can see, the sodium is brought outside of the cell, while the potassium is brought inside of the cells, thanks to the ATP. And this is the basic functioning of the action potential, which, as said before, is at the base of all our thinking, the math, the thinking, our thoughts, and all the circuits that compose who we are, essentially. However, there are diseases that affect the brain and that play and modify this ionic concentration inside of the brain. So epilepsy has been a focus of Dr. Gluckman's research for the past 10 to 20 years, essentially. And epilepsy affects millions of people, as it's possible to see in the graph. And this widespread disease essentially has many barriers, and it does not yet have a cure. And there's no cure yet for epilepsy. However, people can receive treatments, and the symptoms, the seizures, can be calmed down. However, there's still plenty of research because not all these treatments are 100% effective. So another focus of Dr. Gluckman's research that is connected to seizures and epilepsy has been spreading depression or spreading depolarization. Spreading depolarization is an inactivation of large areas of the brain that has been noticed in 1944 for the first time, and that has been observed to be related to seizures as an effect, as like an incipit of a beginning of a seizure. but also in migraines and other diseases. There's plenty of research that is going on in spreading depression, as we know very little about it. So what is happening to the action potential when we're experiencing spreading depolarization? First of all, the concentration of potassium ions in the extracellular spaces increases, passing from 4 millimolar to 40 millimolar. I want to bring your attention to it because now the animation is going to show how it increases. And I want you also to look at the graph and notice how it changes. So as we can see, the concentration in the extracellular space increases. And what happens to the action potential is that it enters in a flat after the sodium has been, the sodium channels have been opened. Why does this flat happen? This is called a quasi-steady state moment for the action potential. And essentially, it happens because there's not enough strength there's not enough current of ions. The current is not strong enough for the ions of potassium to flow out. And essentially, this keeps the potassium channels open, and the cell cannot repolarize in those moments. In this moment, at the physical level, this is what's happening, but at psychological level, we cannot think. Our brain is stuck. There is no processing, and we are not able to do the normal things we would be able to do normally. Think of when you're having a very strong headache that you cannot think at all. So that's more or less what's happening at biological level. So how do we study this more, and how do we know more about this? Current techniques to study the real-time changes in this concentration and in this molecular dynamics have been in the past explored. These are some of them: hemodynamics, metabolic change analysis, electron paramagnetic resonance. However, they all share multiple disadvantages that make the understanding of these diseases more complicated for us. So all these techniques share, like some of them, low spatial resolution, which means that they're too broad in the space. So we are able maybe to see 1 centimeter cube what is going on in our brain in those concentrations, but we are not able to tell what is going on in 1 millimeter cube, for example. So it's very broad, and we're interested in knowing what is going on on the membrane of the cell, you know. Then they all have low temporal resolution, which means that we might be able to know what's going on in the dynamics of the ions, but in long periods of time, such as every minute or every hour. But we want to be able to tell at the millisecond, for example, or at even below, if possible, what is going on in these concentrations. Finally, all these techniques are very expensive, as they require 2-photon microscopy or other very expensive equipment. Thus, our solution has been implemented in 2022, an optical endoscope developed by one of Dr. Gluckman's former students, René Gangerosa, and it's basically an endoscope that brings several advantages. So it brings ratiometric analysis, meaning that the endoscope shoots light to a sensor, and this sensor shoots back some other light, and this other light has 2 signals. One is the reference signal, and the other one is the modulated signal, the one that changes with the environment, with the concentration of potassium or oxygen or whatever other analyte we're interested to monitor. In this way, we can determine using light with very high speed in the millisecond range what is going on at this concentration. Further, we're using a tip that is very small, 100 microns in diameter, as shown in the fiber stub part of the slide. And this gives us very high spatial resolution with more accurate readings. Finally, this whole system is much more inexpensive compared to the ones mentioned before, and can be easily reproduced in any lab. However, the challenge is that we need a coating, a sensor that is able to do what I just explained before. So send back a reference signal and a modulated signal. And so we need something that can be coated on the fiber stub, by the way, made of glass, and that restricts the analysis to the extracellular space so that we do not get wrong readings by knowing also the information inside of the cell. And finally, we want this sensor to be customizable so that it can be applied based on our research interests to other ions or analytes in the brain. Our solution was then to develop silica nanoparticles. Silica nanoparticles that contain fluorophores inside that are analyte sensitive and that can be used as reference dye as well, as shown in the diagram. We choose to make them porous so that the ions can still enter, but the fluorophores do not leach out. And finally, we wanted them to be around 300 nanometers for the purpose of this study, so that we could easily see them and notice changes as we progress in our research. However, in case of commercialization or future research, we would probably go even smaller in size, but we're going to go into that later. So how do we produce the particles? To produce the particles, we decided to use a procedure from the 1970s called the Stober procedure. The Stober procedure has several advantages. It is simple, it is scalable, inexpensive, it uses silica, which is biocompatible, and the reagents are very low toxicity, meaning that this is also a safe operation for a chemist to perform. The size of the particle depends on the ratio of water, ammonia, and an orthosilicate. That in our case, we're using the standard one that was used in the original Stober procedure, which is tetraethyl orthosilicate, or TEOS. And essentially, regulating this tree concentration, we are able to produce particles that are monodispersed, meaning they are all in the same size range with very few variation in size. And we can basically produce as many as we want. So we first add the water and the ammonia, and then we add the TEOS. And then, as I was mentioning before, following this graph, which was the one from the original paper, we determine what size we want of the particles. In this case, this graph is calibrated for 0.28 molar TEOS, but we can imagine that as we increment the amount of TEOS, the particles will be larger. These particles are fully spherical and they are solid, and they can range between 50 nanometers to up to 1 micron, giving us plenty of range to explore. Looking into the reaction a little more in depth. The first step of the Stober reaction is the hydrolysis. Hydrolysis, so this is TEOS, the tetraethyl orthosilicate I was mentioning before. It has an toxic group, or ETO, which is this group. This group reacts very well with water, breaking out and leaving a silanol group instead. These silanol group molecules are very reactive, and they tend to react together, forming siloxane. Siloxane will act as the nuclei for our nanoparticles, essentially. So now I'm going to show you how these particles grow. These particles grow through aggregation. So what you're going to notice is that the nuclei, or the siloxane, is going to turn into a yellow particle, while the silanol will turn into green particles. And you'll see how this essentially changes. So as we can see, the silanol groups are forming inside of the solution, and then they all aggregate towards the nuclei. The molecule starts then growing, becoming a particle, and several other particles start growing, all about the same size inside of the solution. This is what we produced in the end. So now, ignore one moment that I wrote the 5% TMOS. I'm going to explain that later. However, these are the particles, and as you can tell, all these particles at the SEM microscopy, they're all about the same size. And we're talking about 300 nanometers in size. They're fully spherical, they're like beautiful, solid, and everything. And it means that we managed to reproduce the reaction correctly. Going a little more in depth into the data, we run some size distribution analysis, and looking at those three graphs are 3 points that were taken from the earlier Stober graph, and they're varying in concentration of water and ammonia for the same concentration of TEOS. And if you look at the x-axis, that's the particle diameter, while the y-axis, that's the percentage of particle by number. And as you can tell, like looking at the blue line, we have particles that are all about in the range of 300 nanometers as the median. We also try to repeat this procedure using ammonium hydroxide, which should essentially increase the speed of the reaction. However, what we noticed is that ammonium hydroxide in higher concentration makes the particles much bigger, about 100 nanometers in size, which is out of our target. Thus, it also was convenient because ammonium hydroxide is toxic, so we reduced the amounts of that chemical that was used. The particles are monodispersed, meaning again that they're all about the same size, and minimal uses of ammonia were used to produce the particles. So the next step is now adding porosity. We want these particles to not just be fully solid, as I said before, but we want to be able to encapsulate a fluorophore inside of them. Thus, we thought of adding a secondary reaction, not just TiOS reacting, but adding another orthosilicate, which has a methyl group, TMOS, tetramethyl orthosilicate. Having the methyl group, TMOS reacts much quicker, becoming a competing reaction with TiOS. In this way, we are, if you can notice indeed, TMOS is a smaller molecule. That makes the reactivity much quicker, essentially. In this way, what happens to the aggregation reaction is, if we add a mixture of TiOS and TMOS, what will happen is that we still have the formation of the silanol groups inside of the solution. However, this one will aggregate much faster, leaving holes that are going to form the pores. And these are the pores that we want to encapsulate the fluorophore, essentially. The problem is now, how much TMOS should we add in the reaction compared to the TiOS? Running a brief study on the concentration of TMOS as orthosilicate, we noticed that for lower amounts of TMOS, 5%, 20%, no big change, no change was basically noticed in the particles. This made us think that the TMOS was reacting so quickly that it would start creating all the nuclei in the solution. And then the TiOS remaining, being a majority, would start building upon them, closing all the pores and increasing the size, making them basically identical to the 0%. However, from 60% to about 75%, we noticed that particles start decreasing in size. Something is clearly going on there. And finally, above 80%, we noticed that the particles are no more, and what we have is just amorphous silica that is present, meaning that we don't have particles anymore, but the aggregation reaction is happening so quickly that it just became straight up amorphous silica. Looking at some more data, since we cannot rely only on SEM images, we looked at the surface area and the porosity of the particles, as well as pore size and this particle diameter. And the data is confirming basically what we just said. So the particles are basically the same size until 60%. They have the same surface area, and the porosity is basically zero until 60% TMOS. After this, there's a sudden increase that peaks at 75% in the surface area and porosity. You might be wondering, why are we looking at the surface area? Well, if we have a solid sphere and pores start increasing inside of the sphere, well, the surface area is going to increase also in the sphere. And so it's another indicator to confirm the porosity. The porosity in the last graph is the porosity by volume, meaning that we went and measured using pressure and special techniques how much, what is the volume of the pores in the particles, essentially. We also looked at the pore size, which, as you can see, is in the 1 nanometer range, which is very small and might just do the trick for the fluorophores we are going to use. And finally, there's also a decrease that was noticed in the particle diameter, which, I mean, we saw already into the SEM images. Thus, if we look, these are the changes that have been noticed by the SEM. And if we look also on the graph, the 5% TMOS, it basically is unaffected, while when we went to 75% TMOS, there's a drastic increment in the porosity and in the smaller size of the particles. Now that we have our particles that are highly porous and they are able to encapsulate a fluorophore, what's going to happen is that we need to add a fluorophore, and we need to test how these particles actually change the properties of the fluorophore. So going into the adding of the fluorescence, we decided to go and test the particles using fluorescein. Fluorescein is an innocuous and inexpensive dye that we have in large quantities in our lab. And in this way, we essentially added it to the solution by adding it to the ethanol. And the particle, well, they were fluorescent in the end, which is great. However, we had some leakage that was noticed by two ways. The first one, we observed the particles, how their absorbance of fluorescein changed over time. So we observed the particles right after production, they were very absorbent. The fluorescein was very absorbent. However, when we observed them 60 days later, the particles were basically indistinguishable from their control, so from the particles without the fluorescein, meaning that some leakage was going on. Another method to observe this was by observing the liquid around the particles. So what we did was essentially washing the particles over time, and some samples of the surrounding liquid was taken and analyzed by absorbance. So what we noticed that there was a decline in the fluorescein absorbance until day 0, but when we run again the experiment 60 days later and we watched the particles again, there was a sudden increase in the fluorescein in the surrounding liquid, in the waste, in the wash, meaning that the fluorophore was leaching out from the particles. We had to put a stop to this, and what we decided to do was to create a shell to prevent the particles, the fluorescein, from leaking out. And to produce the shell, what we did was repeating the same procedure, the same Stauber procedure after we produced the particles. So let's be clear. First, we produce the particles without a shell. We make sure that the fluorophore is inside of them. Then we take these particles out and we repeat the procedure using the already formed particles as primers for the new shelled particles. And what we notice is that to produce the shell, we varied the concentration of orthosilicate. Essentially, we use very low amounts. And so what we noticed is that until about 0.075, there was no big change in the size of the particle. I mean, that something is happening, but we cannot really see it by the particle size. So looking at the 0.075, there's a double peak. And that warned us that we added too much orthosilicate. Why so? Well, first of all, a double peak means that no more orthosilicate, no more silanol is aggregating onto the particles, but new nuclei, new particles are forming in the solution. And so we have that peak at 100 nanometers, meaning that small particles are growing. However, limited by the amount of TiOs and TMOS into the solution, these particles only stop in size at 100 nanometers. Going even beyond, we notice that this is confirmed. There is a drastic increment of the particles at around 300 nanometers with an increased percentage by number, confirming our suspects. Further, if we analyze the particles under basically absorption and emission analysis, in particular the absorption analysis, we notice this trend. And I want to bring your attention to the starting to rise curve in the absorbance. What is happening here is that the orthosilicate, the silanol groups, are all aggregating onto the already formed particles. And doing so, they're occluding slowly the pores. Since we're using very low concentration of orthosilicate, this occlusion is not enough to fully close the pores. And we indeed have the maximum retention at 0.025 molar, meaning that at this point, we are hitting the sweet spot in the size of the pore diameter, meaning that the pores are so small that they do not allow the fluorophore to go out. But still, there is light that hits the fluorophore, making it be able to be detected, essentially. Before that, the particles are still retaining some fluorophore, but the pore is still too big, and there is some leaching out from them. if we continue on looking on the graph, what happens is that the pores start being filled up. There is too much silanol in the solution, and this essentially is occluding the pores, and it leads to a decrease in the absorbance, which then becomes essentially zero. And this is also confirmed by the emission analysis. To be fair, emission can only be used qualitatively, meaning that we cannot say that the fluorescence of the particles with the best pore size are emitting twice as much as the ones without, but we can see that there is a higher level of fluorescence. So there is for sure more fluorophores into those particles. Going on, this was our choice then to produce this particle using 0.025 molar of orthosilicate to produce the shell in the correct size. Looking at the impact of this research, we need to consider three main factors under the ethical perspective. First of all, this project all follows the responsible conduct of research, and we are basically using ethically equipment and all of this. Then our intended outcome was to improve our understanding of neurological diseases and effects of these neurological diseases on the brain at a molecular level. And this has been met because now these particles can be encapsulated with fluorophores that are potassium sensitive or oxygen sensitive and be able to study how those concentrations change in very small space. And so our outcome is met. Related to the unintended consequences, we made this particle and this device so small that it cannot physically harm the animal. This optical endoscope is made for in vivo studies in the long term, meaning that the animal will be running around with the optic probe into their brain, but this is so small that it will not either harm nor even annoy the animal, and it will basically not give them problems. Looking at the environmental perspective, as I said earlier, the Stober reaction uses very low toxicity reagents, and it basically does not harm the environment, the waste that is being produced, if not in very low amounts. Further, these particles can be engineered for other applications outside of the biomedical field, outside of the brain field. They can be used, for example, for detection of pollutants in the air or pollutants in the water. So it can be used as a general sensor for any other environmental application. Looking at the economic perspective, this production is inexpensive. It's scalable, meaning that it can basically be produced in large amounts without any changes in the reaction, in the yield of the reaction, and basically maintaining a very high profit from it. And the only problem that comes, and the only expenses that come with the production of this particle and this sensor, is that it's very expensive to characterize these particles. Not only because we use the SEM, which is very expensive to use in first place, but also because there's many other steps, such as FDA approval, such as all the BT, BJH, which are all which are all
Is that still too much?
Yeah, and so these are all expensive techniques, right? So if we want to have manufacturable and sent to the industry these particles, we need to consider that and take that into account. However, this is a great candidate for industry application since it can be easily produced, as said before. Going forward, health and safety consideration already exist silica FDA approved nanoparticles. Those are called CDOTs, produced by Cornell, and they've already been applied for biomedical applications, but the difference is they are not fluorescent in the same way that ours are, and also they tend to biodegrade much faster than ours. However, this also this paves the way for us to have FDA approval, since our particles are also made of silica, which is already been shown to be biocompatible and usable for this type of application. Finally, this is a safe process that does not harm neither the researchers nor the environment, as said earlier. Last consideration is the social one. So all these projects is focused with the idea of contributing to the knowledge we already have and that we are lacking of the brain and the better understanding of it. Its purpose is to be used in animal studies so that we can find eventually a cure for all the neurological diseases I mentioned earlier, if not even further others, such as Alzheimer's or Parkinson's, which are still being deeply researched and we still do not have a cure for. And finally, it can also be translated to other applications outside also of the environmental one or the brain or the biomedical one, depending on the needs. In conclusion, we managed to produce the silica highly microporous nanoparticles with fluorophore encapsulated into them. These particles seem to be able to retain up to 60% of the fluorophore that was added, which is great, meaning that we have a possible sensor for our endoscope. Looking at the advantages, these particles are biocompatible, they allow high spatial and temporal resolution, meaning that they're so small that it will allow us to analyze with high accuracy the changes in molecular concentration inside of the brain without inaccuracies. Further, they can be used for multiplex analysis, meaning that multiple fluorophores can be added inside of the particles, and depending on the changes that you make to your optical endoscope, you can monitor at the same time multiple parameters such as oxygen, potassium, but also in the possible future, calcium, sodium, or whatever else you're interested in analyzing in your research study. Also, this is a stable and inexpensive essentially project, looking of course at only the production of the particles, and these particles can also be customized, which I mentioned many times. Looking to the future steps of this research, for sure there should be a study in the future on how the photobleaching rate of the fluorophore changes from when it's encapsulated to when it's not. So what is the photobleaching rate? The photobleaching rate is how quickly the fluorophore decays and stops emitting light over time. We want to be able to use these sensors for long periods of time. So essentially, we want to decrease the photobleaching rate since it decreases the quality of our readings. And we want to study if these particles, by encapsulating and holding in the fluorophores, not only hold it in, but they also limit this photobleaching rate, which is due to the too much energy of excitation of the fluorophore. Also, we want to test these sensors with actual analyte specific dyes. We did it with fluorescein, which however is only pH sensitive and highly unreliable on it. So it would be interesting to observe how this how these particles work with, for example, oxygen-sensitive dyes. Are they still able to detect oxygen inside of the particle or not? And will they work? Those are some questions that come up at the moment. Further, I mentioned earlier the washing step. So while we are adding the shell onto the particles, how we've been washing them so far and removing the liquid outside, we have just been replacing it using ethanol, plain ethanol. This creates a chemical gradient that at the moment in which the particles do not have a shell, it brings to the leaking out of fluorophore over time. And if we want to retain as much fluorophore as possible in these particles, we should consider replacing this surrounding liquid with an ethanol rich in fluorescein, essentially, so that there is no chemical gradient created and the fluorophore will not leach out while we don't have a shell. After that, once we have the shell, the particles will be able then to be used also in ethanol without worrying about the chemical gradient leaching out the fluorophore. Also, it would be interesting to observe fret excitation inside of these particles. What is fret excitation? Fret excitation, said in very simple terms, is the way that two fluorophores, one excites the other one. And this excitation, this radiative excitation, essentially creates a cascade of fluorescence, essentially, allowing us to save on the reference dyes, essentially, not having to have a reference dye for each fluorophore, and kind of concatenating all the emission and absorbance spectra. We are curious to see if this fret excitation is actually possible in first place inside of the fluorophores, inside of the particles. And as the last point, as future research, it would be very interesting to observe how these particles integrate with the endoscope made by René Gamberosa, and see if it actually can work together. So I want to thank Dr. Gluckman that ran me through all of this research and took me in freshman year. And I want to thank also Dr. Adair and Bernie for all their help and for making this possible, essentially. So thank you so much. I am done. Do you guys have any questions?
So in your next steps, you talk about making these kind of customizable, it's customizable to where you can change the analyte, right? Like you can specifically detect certain kinds of ion. What kind of considerations do you have to take into your fabrication process if you change that analyte?
So the good thing about the Stober procedure is that it's not really affected by the type of fluorophore that is being added into it. So there shouldn't be any problem in basically just adding the fluorophores of interest into the ethanol, and that shouldn't essentially change anything about the structure of the nanoparticles. But of course, like proper replication of the experiment should be done beforehand to check if this statement is true, and further characterization would also be needed to confirm each step that the porosity stays the same, that all the other parameters that we saw before do not change as we are adding the fluorophore. Does that answer your question? Great. Any others?
Could it be done, could it be made, or is it already disposable and biodegradable? I'm thinking about, maybe not applications related to medical field, but I don't know, waste, right? Looking at the waste and water. How it could be, user friendly or population friendly? Can it be produced in big amount and I take it somewhere and then just dispose of?
I mean, of course it would need, this is a sensor, right? So it only senses the environment and sends back a signal. However, we would need in that case a system that is able to detect this light from where we're dropping it and analyze it. But if any engineer is able to design a system similar to the endoscope we have, just for other purposes, we would be able, to apply it, for example, for waste disposal, as you said. Does that answer your question?
Well, I think part of your question had to do with non-silica-based nanoparticles.
Yes.
So, do you know of non-silica-based nanoparticles or porous?
Yes. So this procedure has been done with silica, but it has also been done. We got inspiration also from Dr. Adair's previous research. He made it with calcium phosphate nanoparticles that basically follow a very similar procedure to this one, and those ones essentially biodegrade automatically after they've been applied. They were meant for drug delivery, so that's why. But I'm sure they can be also re-engineered. I'm not sure about the FDA approval related to those, but since they already have been applied for pharmaceutical use, I'm pretty sure that that already paves the way for it to be applied.
Good. Thanks.
You're welcome.
One of the easier to use now.
Yeah. Yes. What are you going, I mean, I know you applied, right, for grad school here.
Mhm.
Are you?
Actually, I am going next year to PhD at Johns Hopkins.
Oh, okay, perfect. So, yeah. You're losing.
I'm used to see new
Oh,
advisors.
absolutely, absolutely. But it's always, I mean, it's
But
great.
he's,
Yes.
he's gonna go work for a colleague of mine.
Mhm.
He called me up and says, why aren't you keeping him?
It's
So,
it's-
because he needs to go
yes. No,
learn
it's
something
great.
new.
It's great, it's great, it's great. I just know because I remember your name.
Any other questions?
You guys are really quiet. Ask questions.
Chemistry scares people. I heard that.
Like, chemistry isn't really my strong spot, but
Can I ask him about the neuroscience part?
give me a second.
Please don't ask me if we are in a simulation. I do not know.
So I know you talked a little bit about like the sensors into the putting sensors into animals and making sure it wasn't harmful and stuff. Like so, being able to sense whatever you wanted. Would this device like so if we're talking about animals, like if you wanted to experiment on say an animal running, would it be hard to make sure that this device stays intact and like these particles don't get affected by general use in the body? Or
I mean, that's a very good question. So, in our studies, we keep the animals inside of specific cages, so there's not much space for them to run. However, we also keep them with like a cord attached to their top, and essentially, regardless of where they move, we have also like a small like pet that we give them that keeps it in place and allows only the cord to move and not the sensor to move around and start digging in the brain, you know.
Yeah.
So that prevents it. There's a precaution that we use for not the animal prevented from hurting the cell, essentially.
That's not right. We're doing mice studies now. The mice run about, when we let them, they run about 2 kilometers a minute.
It's faster.
What's that?
No, I think it's faster than light. I mean, it is what, 7? No.
They do that over about 6 hours. So
All right.
almost continuously, so it's just.
Do we have any question from the Zoom people? I'll take that as a no.
All right, well.
Well, thank you so much for coming to my defense. I'm very happy.
Hold on, you got a chat, Alessandro.
Oh wait, it's probably the where do I see it?
When you chat.
Click on
Yeah, but I
chat.
Click
cannot.
on chat.
Click on chat and then.
See where it says chat at the top?
I do, but it does not.
You think you
Oh,
have to
because
get
you
out
have
of
to
your
turn off the
PowerPoint.
laser pointer.
Yeah. Okay. Where we go? I'm coming on. It's just not appearing for me.
Just give me a second, I'll open up the Zoom meeting. and go from-
Yeah, thank you.
You might have to, I think it's a second desktop. You might have to,
Maybe
Yeah,
it's
it is. Don't worry, I'll have it open in a second.
The projector screen is like a second screen, so you have to scroll from your computer up to it, but the chats are congratulations and then the evaluation form.
Oh, okay. Good.
Well, there you go.
it works.
So yeah, that works great.
Yes.
Well, thank you then.
And I can't even see the chairs.
So it's not just, okay, good. All right, and they cannot even log out, so.
Okay, so stuck here forever.